Our second presentation this morning is from the recipient of the ACCP Fellowship Award. The ACCP Honorary Fellowship Award is given annually to a non-member of ACCP and is meant to recognize primary activities within the immediate domain of clinical pharmacology. The award recognizes overall contributions to the field rather than for any particular scientific work by a senior investigator or authority having a national or international reputation in the scientific, public service, legislative, governmental, or other area of endeavor impacting the field. The recipient of this award is Dr. Ted Grasela. Dr. Grasela is a scientist, entrepreneur, and systems architect. He received his PharmD in 1979 from the Philadelphia College of Pharmacy and his doctorate in epidemiology from the University at Buffalo in 1999. He is currently an adjunct professor in the Department of Pharmaceutical Sciences at the University at Buffalo, and he previously served on the faculty of the university, where he was an associate professor of pharmacy, chairman of the department, and director of the Center for Pharmacoepidemiology. Dr. Grasela co-founded Cognigen, now part of Simulations Plus, and initiated the Pharma of the Future program. Through his pioneering work in infrastructure development in population, pharmacokinetics, and pharmacodynamics, Dr. Grasela was a leading member of the first generation of scientists who moved clinical pharmacology applications from individuals to populations of patients. He has further expanded quantitative assessments of clinical pharmacology in the area of outcomes research, safety assessment, and pharmacoepidemiology. Dr. Grasela has an extensive list of publications. He was recognized as a fellow of the American Association of Pharmaceutical Scientists in 2008. He won the Gary Neal Prize for Innovation in Drug Development in 2013. And for 30 years, he's been a recognized leader in clinical pharmacology. Welcome, Dr. Grasela.
Hello, everyone. I would like to thank the members of the awards committee for this recognition. Whenever I have attended one of ACCP's annual meetings, I was impressed with the intimacy and congeniality of the assembly, and I'm honored to be welcomed by the society in this way. This award recognizes overall contributions to the field of clinical pharmacology rather than any particular scientific work. This had me thinking about my work over the past 30-odd years and how they add up to a career and culminate in being honored with this award. In truth, it has been more like the punctuated equilibrium described in evolutionary science, a series of episodes of rapid advancement intermixed with periods of wandering about trying to figure out what just happened and what to do next. And as I think back on my career, most of the fun has been in the periods of wandering about. My latest experience with this punctuated equilibrium phenomenon has been through a series of projects in global health. These have been transformative for me, not just because of the scale and complexity of the challenges I've encountered, but also in the potential for new tools and processes for harnessing collective intelligence and working constructively with this complexity. Today, I want to share some observations stemming from this work and offer some comments about a pathway to innovation in global health. By way of an introduction to this, I'd like to present three key ideas and have you consider them in setting the stage for this presentation. First, the challenges in global health are incredibly complex with multiple interacting risk factors. And this complexity often renders top-down solutions inadequate and prone to failure at scale. Second, there are ways of working constructively with complexity that involve coupling creative thinking with new tools and knowledge management processes to address seemingly intractable problems. The tools and processes can expand our awareness of the context surrounding these challenges and help guide our actions to the extent that we cultivate our ability to tolerate the ambiguity and uncertainty that accompanies an immersion into complexity. Lately, I've been playing Nintendo's Mario Kart with my 5-year-old grandson and getting a lesson in humility. He's not very patient about teaching me the game, and when he gets far enough ahead, he actually stops his car and encourages me to hurry up so that he can make it a closer race to the finish line. My problem is that I struggle to get a feel for steering. I'm constantly bumping into walls or falling into the abyss along the racetrack, and you fall further behind with each of these interruptions. My grandson, on the other hand, pretty much stays in the middle of the lane. His velocity made good is much better than mine because he doesn't make the unforced errors that constantly slow me down. I first learned about the concept of velocity made good, or VMG, in sailboat racing. A sailboat can't sail directly into the wind, and VMG indicates the vector of the speed towards the direction of the wind. Now, if the direction of the wind is aligned with the location of your destination, your VMG might be quite slow. But it might be enough to win a race over others who are unable to make an optimal VMG. I believe that the concept of velocity made good is an apt metaphor for biopharmaceutical R&D and is useful in thinking about approaches to the challenges of global health. In pharma, by virtue of long-standing regulatory requirements and well-accepted patterns of research, the R&D pathway for new medicines follows a prescribed pathway from discovery to market authorization. This prescribed sequence of studies and decision-making milestones brings discipline, transparency, and accountability to the R&D process. It is optimized to maximize the velocity made good, because regardless of whether a drug candidate succeeds or fails, the requisite information to approve or disapprove further investment is available at key decision-making milestones. This concept of VMG is at the core of the pursuit of getting better at getting better. Learning how to make progress against difficult challenges without making unforced errors that waste time, money, and the opportunity to advance innovation. The concept of velocity made good in global health research is more complicated than pharma for reasons that have to do with the complexity of the challenges. The Sustainable Development Goals were adopted by all United Nations member states in 2015. The 17 ambitious goals recognize that ending poverty and its associated adversities must also be coupled with strategies that improve health and education, reduce inequalities, and spur economic growth, while also addressing climate change and preserving the global ecology. The complexity inherent in working to meet these goals originates in the wide scope of interconnections between these challenges. The complexity is shaped by a myriad of dynamic factors operating at the individual, community, societal, and global level that can lead to the emergence of unexpected conflicts and unintended outcomes with any proposed solution. Understanding the context of the challenges at each of these levels is vital to finding successful, scalable, and sustainable innovations. The fundamental obstacle in tracking the relevant context for global health research is that the problems encountered are often complex, interrelated, and not well defined. This slide by Sarah Jensen and her colleagues describes the effects of poverty-associated adversity on structural and functional neurodevelopment in children. The overlapping and interacting sources of complexity create tremendous uncertainty in the development of an action plan intended to provide insight and traction for a candidate innovation to impact neurodevelopment. While the strategic rationale for addressing a specific problem may be well supported, the efforts to design and operationalize an action plan often generate innumerable questions about the specifics of the work and its likelihood for success. Research programs in global health are conceived and designed by independent academic researchers along different disciplinary lines. The studies consist of different study designs, different target populations, different inclusion and exclusion criteria, different endpoints, and so forth. So on the one hand, these varied research programs create a rich ecosystem of hypotheses, results, and ideas that provide a glimpse of the many dimensions of the underlying complexity. On the other hand, there is a lack of coherence across these multiple independent research projects that makes it difficult to integrate results across studies and effectively guide future research planning and policymaking. While we may get hints of progress in understanding complexity and context, the velocity made good for these programs can be disappointing. Think about the many conceptual schemes like this one that you have seen. Think about how they become a static work of art rather than a dynamic living document. Imagine the value if we took these schemes as starting points and made a societal commitment to continually refine any imperfections as new knowledge emerges. One way to begin understanding the complex systems inherent in global health is by describing them in detail, mapping out the many parts, the multiple interactions, and how they change through time. This slide shows two examples of a conceptual scheme. Shown on the left is a scheme describing our current detailed understanding of the life cycle of the Plasmodium parasite. It has been 124 years since British scientist Sir Ronald Ross discovered that the female Anopheles mosquito transmits malaria between humans. This finding provided the foundation for scientists across the world to better understand the deadly role of mosquitoes in disease transmission. The different entities on this scheme and their causal relationships have been realized via constant iterations based on the emerging science in entomology, biology, pathophysiology, and epidemiology of malaria. And we are still learning more as the technology evolves for measuring different forms of the parasite over its life cycle. On the right is a conceptual scheme providing a map of hypothesized pathways to environmental enteric dysfunction. This is a common but poorly defined disorder of the small intestine among adults and children living in low-income countries, particularly where water, sanitation, and hygiene practices are suboptimal. Environmental enteric dysfunction is characterized by altered gut structure and function, leading to reduced absorptive surface area and impaired intestinal barrier function, along with chronic systemic inflammation that leads to a wide range of adverse effects. There is currently no accepted case definition or gold standard biomarker for EED, making field studies challenging and complicating the effort to provide adequate nutrition to affected populations. Although the scheme on the left is more definitive, both of these provide a structured distillation of extant knowledge from the field and represent attempts to get a picture with a high signal-to-noise ratio. These schemes bring parsimony and transparency to the collaborative process and assist in exploring the boundaries of disciplines and the space between them to identify knowledge gaps and help design the studies to fill these gaps. Of course, there's nothing new about the role of conceptual schemes in model-based drug development in pharma. Louis Shiner's landmark publication on the learn and confer paradigm emphasized the importance of learning as much as we can about the determinants of drug safety and efficacy, and turning that knowledge into mathematical constructs to perform simulations that give insight into the likely outcomes of clinical trials. At the time that Elion and Lewis published the paper on the right, the concept of an effect compartment was quite novel. Compare this scheme, though, to the current models in systems pharmacology. We have come a long way in our thinking. As we've seen in the examples above, conceptual schemes are commonly used in global health research, but we need to take these to a higher level of explication with much greater and more intense participation across disciplinary lines. We're going to need conceptual schemes that involve an ongoing systematic effort to engage with a broad network of experts and disciplines and reflect the complexity and ambiguity of the challenges they seek to describe. They must represent the key ideas, hypotheses, intuition, and knowledge relevant to the challenge at hand. They must be continually updated and refined as new knowledge and ideas emerge. And they must be employed as a basis for performing a question-based review at key milestones. This would allow us to assess program progress, maintain transparency and resource decisions, and avoid premature endorsement of poorly conceived action plans. The creation and vetting of conceptual schemes in a communal setting will require new tools and processes that foster the systematic acquisition and integration of new knowledge into knowledge graphs, the assembly of the relevant knowledge into conceptual schemes, and a review process that can enable the vetting and adjudication of conflicting ideas while communicating iterative improvements to the community for feedback. We are experimenting with some of the early generation tools to facilitate cloud-based knowledge management and collaboration activities. And we have found them to be quite promising. And we expect that the tools will continue to evolve and have a profound impact on a new approach to problem solving that leverages the science and processes of collective intelligence. This is a snapshot of the knowledge graph that we have been working on in global health. My colleague David Price coined the term Thriveome to refer to a collective of such knowledge graphs generated by research teams working in global health and beyond. The Thriveome includes the body of questions we have been pursuing, extant knowledge about risk factors and their role, results from data studies, and outcomes of attempted interventions. Each of the dots on the graph represent a node, a specific bit of knowledge or data. The lines indicate multiple interconnections between these nodes, and the colors indicate different areas of focus. Each item on a conceptual scheme is a node on the knowledge graph. The conceptual schemes show the relationships between nodes in a particular section or space within the Thriveome. This is analogous in a way to naming the constellations in a night sky crowded with stars. The mapping of the constellations helps us to make sense out of the complexity above us. The idea of the Thriveome builds on the concepts and goals of knowledge management from the early days of computers and the internet. Visionaries like Doug Engelbart and Tim Berners-Lee saw a future where knowledge was easily and freely accessible. And to some extent, this future has arrived. Googling, the act of querying the Google Knowledge Graph, is a common pastime. One can easily book a table at a restaurant, but of course it is one thing to check on the opening times of a restaurant. It is quite another to find a new medicine, a vaccine, or a healthcare policy. As we envision it, the Thriveome would be used by scientists, governmental agencies, funding agencies, and policymakers to immerse themselves in the complexity of a challenge. This immersion would enable the visualization of the different dimensions of the challenge, accelerate recognition of connections to the adjacent possible, facilitate the identification of relevant domains that need to be further explored, and provide support for specifying requests for information and requests for proposals that generate the knowledge for iterative refinement of conceptual schemes. The 21st century could be a great age of new commons. And unlike the tragedy of the commons, which often occurs when unbridled access is provided to community property, it is not possible to use up the ultimate commons, information and knowledge that can be used to help solve global health and development challenges. If we were to match the growing capabilities of our technology for managing and sharing collective intelligence with comparable social and organizational imagination to use this intelligence, we could confront the complexities we face and seek out truly impactful innovations. Ernesto Ceroli has worked for almost 30 years on local economic development in Africa, Australia, and North America. He has a wonderful story about his early days working on economic development in the Zambezi, and you can find it on YouTube. Briefly, the Economic Development Group commissioned by the Italian government decided that a parcel of land was incredibly fertile for growing tomatoes. As much as they tried, the Italians couldn't get the locals engaged in the effort. So the Italians decided to do the project themselves. After a few months, the tomatoes were ripe, delicious, and ready for harvesting. One morning, the team went to the fields and found that they had been destroyed. All of the tomatoes were gone. Shocked, they went to the villagers and asked, "Who did this?" And the elders replied, "The hippos. That's why we don't grow food on those fields." When asked why they didn't tell the Italians that before all the work, the elders replied, "You didn't ask us." Mr. Ceroli tells that story to remind you to work hard at understanding the local context before you come up with a top-down solution. Risk embarrassment by targeting the wrong problem. We become so sure of our expertise and our beliefs, and in our eagerness to find solutions, we don't take the time to truly understand the issues. Impatience becomes an obstacle to achieving an optimal VMG, particularly when a solution this setting is more than a narrowly defined goal. It is likely a set of coherent actions that seek to amplify positive inputs and dampen down the negative factors in a larger context, so to avoid unintended consequences. One of the complicating factors in trying to make velocity made good in Mario Kart is that there are many different tracks to choose from, and the number of choices and their complexity continue to multiply. The situation is familiar to global health researchers. Each challenge can feel like a different racetrack, and the more we learn about each challenge, the more we are struck by the previously underappreciated complexity. Any VMG that we make in thought, theory, or practice innovation will be enhanced by evolving maps of the routes we need to navigate. Drawing on the science, tools, and processes of collective intelligence, we can create the conceptual schemes that can be the source of those maps. The schemes become a navigational aid to reasoning cogently, coherently, and collaboratively in the present day. And as they mature over time, similar to what we have seen with malaria, they constitute a multi-generational gift, providing an ever-expanding knowledge base for researchers of the future. I'd like to thank a number of my fellow navigators on the journey through ambiguity and uncertainty, including my grandchildren, who continually remind us that finding our way is a lifelong journey. Jill Fiedler-Kelly and Cindy Wallawander, and the scientists and staff of Cognigen, Charles Dement and Robert Slusser, who served as mentors and colleagues on the Pharma of the Future initiative, which continues to shape my thinking today. The global health scientists and clinicians with whom I have the pleasure of collaborating. And last, but certainly not least, I want to thank David Price for his guidance and patience as I came up to speed on the tools of collective intelligence and the possibilities for artificial intelligence to help craft solutions to the challenges of global health and development. Thank you very much.
Hello, everyone. Welcome to the ACCP annual meeting. It's my honor to moderate the question and answer for the talk of Dr. Ted Grasela. Just a little reminder, please use the Q&A window that you have to enter the questions and the tab to chat with other attendees. I welcome all the questions that you have for Dr. Ted Grasela talk. So far, we don't have any questions. I do have questions for you, Dr. Grasela. So the title of your talk is about velocity made good. I have to say that I learned this concept today. And I would say that it's a general concept that's not only applied to science, but also to personal life. So how can you really tell if you are on a good trajectory? Can you tell us?
Thank you, Sam. Thank you for moderating this question and answer period. You know, oftentimes it's harder to know that you're on a good trajectory, but perhaps easier to know that you're on a poor trajectory. So in science or even in personal life, it's interesting that you made that connection. A lot of times we don't like to ask certain questions that are troublesome or embarrassing or reveal maybe our insecurities about what we do and don't know. And if you find yourself working with a team, for example, and they're reluctant to address what if questions, you know, so you formulate an action plan and then you say, well, someone comes in and says, well, what about this? And what about that? Or what if this happens? If you find that the team is reluctant to pay attention to those things, I think that's one of the clear signals that there's a problem or potential problem with the trajectory, because it means that there are a lot of issues that may very well be relevant to making corrections to the action plan that people are not paying attention to. And that's where I think we get into trouble with unintended consequences and difficulty in pushing innovation along.
That's outstanding, actually. Yeah, and it's a general concept, actually, applied to science and to life, as you mentioned. So I don't have other questions. Please, attendees, if you have any questions, don't hesitate to drop them on the chat box. So I do have other questions myself, actually. So your talk, Ted, emphasizing on the importance of the conceptual schemes in developing and understanding the complexity and the context. In reality, how is this different from the usual practice of science?
So, of course, conceptual schemes are a really important part of science. And whenever we work with clients or struggling with a particular question, I like to recommend that people always go back to the current picture that we have that represents our best understanding of a particular problem that we're trying to solve. And so that using that as a basis for conversation and for discussion about what do we really know versus what we think we know can be very informative. One of the key challenges that I think we have in science with these conceptual schemes is that they take on a life of their own. We get to be overconfident in the schemes and perhaps fail to recognize its limitations or places where it might be incomplete. So the schemes become a justification for an action plan or for a research and development program, rather than an opportunity to ask questions about what maybe we missed and don't know that we ought to be able to take into consideration. It's always interesting to me about, if you think about drug development programs, inevitably the conceptual scheme that was developed early on is a slide that you see in every presentation that happens about that drug. And it makes its way into these presentations. And the challenge is that if there are issues with that scheme, we may not know about them. People may not have paid attention to them. They may have been reluctant to look into those issues until the problems arise in later stage development. So I think that the usual practice of science is focused on these schemes. It's just that we don't take the time to continue to question them.
Absolutely, yeah. And I'm looking at the questions. I think I haven't gotten other questions, but I do have a third question, actually. I really loved your talk. And so my question is, you mentioned also in your talk the importance of an immersion in complexity using a new concept to me, which is I hope I pronounce it correctly, the Thriveome. So can you say a little bit more about this and is it really relevant to the pharma R&D? It's a very new concept for me, so I'm learning.
Thank you for that. And you got it close. It's Thriveome.
Thriveome.
So if you think about the conceptual scheme being a distillation about what we know, right? And so we're really trying to focus on the signal, and we block out a lot of the noise so it becomes easier to tell a really well-crafted narrative about how a drug works or a particular situation that we're dealing with. The Thriveome, as we envision it, contains all of the loose ends that we may have encountered during a drug development program, but didn't really find a place to fit into the narrative. Maybe it speaks to some potential pharmacologic effect of the drug that's off target, and maybe we don't really want to chase that down very much. So later, when we're looking at all of those loose ends, the Thriveome is the place that connects all of those loose ends to the core conceptual scheme that we're working with. So you say, geez, I think that there might be some issues with inflammation with the COVID vaccine. Well, what do we know about the relationship between inflammation, COVID, and the vaccine itself? What are all of the extra dimensions that maybe somebody has hypothesized or thought about, published some email or an idea? Those kinds of pieces of information become accessible to you within this knowledge graph. It's those places that you go for inspiration and maybe for a new idea for how you can investigate something that wasn't properly investigated in the beginning. So in the end, what it represents is the place where all of the information is accumulated, not just the information that supports a narrative.
That is outstanding. I think we are on the top of the time. Thank you so much, Dr. Grasela. That was an outstanding talk, and congratulations again on your award. Do you have any last final words to our attendees?
Well, I see that we're out of time, and I just want to say thank you for your attention to the presentation and for your questions. Thanks very much. And also thank you for the award. I really appreciate that recognition. It was quite a surprise, quite a pleasant surprise. Thank you.
Thank you, Dr. Grasela.